Awesome. Awesome. Thank you very much. Really excited to be here. We're really happy to sponsor this event. So I'm Andrew, and then we have a few people from Future House that can maybe help advise if you have some questions about biology. So where's Future House? So they're in the back here. We have Mike, who's right there. Mike is an expert in proteins and laboratory experiments, genomics. James is the best Python programmer I know in the world. And then, is Manu back there? Oh, Manu is here. Manu is an expert in protein design. I don't know anybody better at protein design. And then Sam is somewhere, green hair. Oh, Sam's hiding back there. Sam C, who the first author on ChemCro paper, expert in chemistry and using agents in science. We are going to join projects. Love to help you guys. Any questions about biology if it's new to you. So I'm just going to give a few slides about Future House. So the last 10 years have seen some incredible advances in biology tools. So the cost of experiments is getting lower and lower in biology, and also the pace of this is beyond Moore's law. So able to synthesize sequences at like $1 a sequence and sequence them for very low prices. So we're reaching a point now where the limiting factor is now like human intellect to make biology go faster, not any sort of experimental method or waiting for any waiting for experiments to be done. So we think that there's going to be some sort of breaking point now where experiments are automated and quick enough and large enough batch size that we need to start coming up with ways to generate good hypotheses faster and analyze the data faster. So the Future House mission is to build an AI scientist. That is, we're going to build the tools, the laboratory capability, the robotics, and the compute in order to completely automate the scientific process from beginning to end to build a closed loop system that can make new knowledge. So some of the things we think about: literature search, hypothesis generation, automated analysis, a lot of this with both machine learning and language models and agents. So key facts about Future House: we're a nonprofit, we're based in Dogpatch. We actually are trying to build a community space. It's still in progress, but we're going to be hosting hackathons and doing events in a warehouse in Dogpatch soon. Looking forward to seeing you all there. We started in November 2023. We have a wet lab backed by Eric Schmidt and other funders. And we have 15 people and we're growing. So if any of you are interested in applying your engineering skills or your biology skills in an environment like this, please talk to me. Oh no. I had a video of robots doing stuff, but I guess I didn't get the access, so we'll have to skip that.
You can do it by charades.
Yes. So I just want to mention, so basically our goal to automate science is we're building these agents, these kind of like scientific agents, we call them crows. So there's ChemCro, which maybe you've heard of before, which is able to do chemistry with robotics and machine learning models. WikiCrow is able to go through the scientific literature to summarize knowledge, synthesize knowledge, and write Wikipedia articles or answer technical scientific questions. So these are two public projects that have been announced. So we have a Discord. You guys can take a picture of this, and inside of the Discord there's WikiCrow, and I thought like as a resource for this, if you need questions about biology, you can ask it questions. Like I asked it, can I grow extra digits with genetic engineering? So if you want to have 6 fingers, which would be very valuable to get away with crimes, because everyone will think that you're a hallucination, in a Midjourney image. So you can get that done with genetic engineering, and it gives you back like a paragraph of text with citations telling you which specific pages to find in the scientific literature to answer your question, and it's hallucination-free, guaranteed, or your money back. So please join. You can play with the Discord bot. I'm sure it will break, but if you don't know anything about biology, this is maybe a good place to start by asking it random questions. Okay, cool. That's it. I think there's been great ideas in the Slack. Join the Slack. People put up awesome ideas. I don't want to try to give any more ideas, but I'm also looking for a project to join and participate in. So thanks, everybody.
Oh my God, the link broken. Amazing stuff from Andrew. James, GP at DCC. Give us a bit of a talk on DCC.
Yeah, sure. Hey everybody. I do not have any slides, so I'm just going to tell you a little bit about DCVC. We're a early stage venture fund. We focus on the seed and the Series A as entry points. Tend to write $1 to $5 million checks at the seed, 5 to 15 at the Series A. I've been really active in the AI, I'd say, plus biospace. We invest kind of all along the process from companies that are working on computational discovery of new drugs. So that would be companies like Recursion Pharmaceuticals. You may have heard of them. They're currently a publicly traded company. about $2 billion market cap. NVIDIA just invested around $50 million, helping them build one of the largest compute clusters to focus just on discovering new molecules. We led their Series B back in 2016, 2017. We're also investors in a company called Accelera, also a publicly traded biotech now up in Vancouver. They were a company that brought to market the first COVID antibody during the pandemic. So not only on the drug discovery part, but we also invest in people that are applying AI to other parts of the stack. I'd say companies like Unlearn, which you guys may have heard of. They're applying both machines for developing digital twins for patients. So if a patient shows up, how can we predict how that patient might progress without any kind of intervention? And so then we can actually, when we give them a drug, we can compare how that prediction compares to this intervention, and hopefully use that for better, faster clinical trials. And we also invest in things like diagnostics, so really all along the stack of drug development and discovery. Actively managing around $4 billion at this point. We've been around for 10 years. And we'll go as early as founders and pitch deck. So if any projects come out of this, or you guys are working on companies, you just come find either me, my colleague Emily, you want to raise your hand. And I have one more, Richard. I don't know if Richard's shown up yet. We'll be one more person from DCVC here wanted around at some point. Yeah, so feel free to come talk to us, tell us what you're working on, or again, get in touch afterwards if you're working on the company. Thanks, everybody.
Thank you, James. So Don from Upstage, yeah, do come. Upstage has a document analyzer, which you can use in your project to evaluate research papers or others. Use slides.
All right, so who's ready to have fun today? Yeah. I know this is a hackathon. You need tools to build whatever you're trying to build, and we are going to suggest some tools that you can actually use today. So we're called Upstage, and we are a B2B AI startup based in Seoul in the Bay. We're currently in the Series B round. We've raised up to $100 million now, and our flagship products are LLM and Document AI, which means that we will be providing LLMs and Document AI tools today. And our customers range from SMBs to enterprises in finance, legal, bio, and other domains. So my name is Lucy, and you can probably find me around that white couch. We have some t-shirts and stickers to give out to you as well, so if you're interested, you can stop by. So we are building a full stack LLM. So what we mean by that is when you have structured data and unstructured documents, we can process all of those data and put them into RDB or NoSQL databases or vector databases, integrate a generative model, and get responses. Responses can be either summarizations or question answering. And this is a very complex pipeline, but let's get a bit simpler. We want to build demos, applications today, and so I'm going to prepare a brief demo for you. So let's talk CRISPR first. For me, I know a bit of AI, but I'm a total novice in bio. So I wanted to know what CRISPR is, and in that sense, I want a bio tutor built from LLM. So let me introduce you some methods that we can do this, and also introduce some tools on the way. So the basic way is to just bring an LLM and just ask the LLM what CRISPR is. So you can ask questions to Upstage Solar, that is our LLM. And it is very simple because we just integrated Solar into LangChain. And calling LangChain would be just two lines of code actually. And it will answer your question, what is CRISPR in layman's terms, into blah, 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 just using the LLM. You want to use Solar because it's quite powerful compared to other open sources out there. It is stronger than Llama 3 and actually very comparable to GPT 3.5, but much faster and much cheaper, about 3 times faster. The second method would be probably when you want to condition your question on a document. So you probably have a lot of bio documents out there, and you want to ask a question towards that document. But the problem is that your document from the web probably has multiple pages. It probably has a lot of columns, maybe some figures and tables inside. That's why, but that has a problem because it is too complex for an LLM because, as you know, LLMs can only handle text. So that's why you want to use a PDF to HTML parser. We call it a layout analyzer. And you want to convert the PDF to HTML, whereas HTML would be all text. So this is also very simple with LangChain as well. You can just use a Upstage layout analyzer loader and then put in your document, and it will just parse your document automatically. You want to use this because the performance is pretty high. It's one of the top in the classes. And it particularly excels in extracting complex tables. So like this is a typical table you will see in academic papers. And academic papers have very, very complex tables, right? They have merged cells and very hierarchical headers. And our layout analyzer can process all those tables very fluently. And then you can ask Solr to do document QA on this, on the results of the layout analyzer. And then there's another thing that you want to check. Like when you ask a document, when you ask a LLM questions about the document, you always get curious whether the LLM said the truth or not, right? So like when you ask names, what are the names from the paper, and the LLM says something. You want to go and double check whether those names actually exist in the document. But this groundedness checker actually does that for you. So if you input the context and the answer, then it will say whether this is grounded to the document or not. So if you want to check whether the answer complies with the document, you probably want to use this. The final method would probably be to use a vector DB. Because sometimes you not only have one short document, you have multiple documents or very long documents. In that case, LLMs can't ingest those documents because it goes beyond the context length. So in that case, you want to use a embedding model and a vector database, and that is also very easy to use. I will give this code out to the Slack channel so that you can all get a quick start. So retrieval is under 10 lines of code, which will get you started very quickly. So I hope you had fun building Rise apps today with the Upstage full stack. We are giving out free API keys. They will be unlimited, so you can spam it. Our engineers will take care of all the loads. There is a QPS that you need to be aware of, but I suspect it would definitely cover today's needs. Right.
What's the max context window?
It's 32K.
And the output?
32K includes the input and the output.
Oh.
All right.
Fantastic. Thanks.
All right, thank you.
So raise your hand if you've written a research paper. Wow, okay. So all of you have gone through this experience of having some research result and needing to write it up and communicate it to your scientific friends. And typically that's a really painful and laborious process. So I'm going to give you an idea for how you might write your research papers in the future. This is an example of a paper created yesterday about our new BCI dataset release. It's flush with dozens of accurate citations and relevant mathematical analysis. The set citations that can be seen here, and you can see the quality of analysis here. I put a description of the work that we did into the Omniscience paper generator by going to omniscience.tech/research, adding the description to describe paper, and adding the authors and our institutions into the author space. And click write. This paper was emailed to me in less than 10 minutes. The email contained the PDF of the research paper, as well as the LaTeX so that our team could start editing. We used Overleaf and added the style file Omniscience sent to us. And just like that, we were off to a running start with our research project. To use it, go to omniscience.tech/research. We give you a generous 2-week free trial. Thank you.
Does it automatically download data sets that it needs?
So it is integrated with a huge number of terabytes of research data, but that data is basically being continually downloaded in the background. It doesn't download anything that's specific to your paper.
Can you control F the word delve?
Yeah, no, there possibly are some statistical sort of distribution frequencies. We are actually doing research into how to make sure that the word distribution doesn't differ in any way from the existing research corpora. But yeah, I think that last checked, 18% of the sentences in computer science related papers on arXiv were judged to be written by a generative model. So we are already at almost 20% AI when it comes to paper writing. And it doesn't seem like that's going to end. My sense is actually the quality of the writing will get to a point where it would be an unusual choice to like write the entire paper by yourself. Similar story for your granting process. So actually some of our most intrepid users are writing their grants by feeding their history of research to the paper generator and creating basically like 100-page grant applications as a function of their work. Yeah, and I expect we'll like figure out the question of delve and other words that identify generative modeling pretty soon. Good. Any other questions? Okay. So yeah, I guess we are going to give all of you a link so that you can freely use the paper generator throughout this hackathon. And I will probably take all of your presentations and generate papers on your behalf just to sort of just to demonstrate the space of possible research that could come out of a single day. So that on a single day, you can have a hackathon where you produce some project. It has a GitHub repo. Maybe you grab some of the code and you have a language model describe it. But basically, everyone can ship both a project and a paper describing the results of the project in an 8-hour block, which is a solid bit of productivity for a day. So it should be fun. Right, next question. Okay. So we're moving to project pitches from you all. There'll be a timer, so it'll basically be about a minute to a minute and a half per pitch. And we're starting it off with Miles from Cofounder. Miles, are you?
Okay, I guess I missed.
And feel free to keep putting in your projects in here while people are talking.
So, yeah, you'll have about a minute to pitch.
The link's on the slide.
If you want to add your project, I sent it out over the particle about 20 minutes ago, and it's also in the Slack. So do add your project here so that people know your name, they know what project you're working on, and they can find you. Let's hear from us.
Okay, I am Hackathon project. I'm very interested in genomics, and how we can create kind of genes that inform the output of LMs and the decisions would be a kind of selection, basically a population of multimodal models that are outputting a kind of phenome, and then looking at the rest of the phenomes in the population, and then finding mates, and then producing offspring, and seeing how that evolves. A lot of directions for that. I also put on there, I was thinking about leading kind of a Twin Peaks cleanup. I've got some garbage bags and gloves. Maybe during sunset or something, if people want to take a break, I'll probably just go up there, and then if anyone wants to join. Actually, it was kind of a funny coincidence because I saw this morning people already, it was looking really clean the last time I saw it. And if people are actually just doing a cleanup together AI, or together SF, but still more to clean. We could make it perfect. Like we could literally make it perfect. We clean up every single cigarette book. It's already pretty clean. So yeah.
For context, for those who don't know, we are a 3 or 4 minute walk from Twin Peaks, which has glorious views, but typically is tarnished by people who thoughtlessly throw things, throw trash away. So, a lot of appreciation. Okay, Ash is going to give us a presentation around strings for genomics.
Hey guys, my name is Ash. Some of you know me as a person who likes building crazy large search systems. For today, I thought we have like 6-8 hours. What is the biggest thing we can do? I thought we can buy humanity 7 billion days with better strings for genomics. For less Boston, more SF mindset, this is buying $100 billion of value because like the average income per capita in the world is around $5,000. And if we add 7 billion days, this is about 100 billion of value. The way we're gonna do it, one of the open source libraries I maintain is called Stringzilla. It's one of the fastest string processing libraries out there with a lot of assembly tricks. And a lot of researchers worldwide use it for large data processing. But one of the coolest things that it has is like global alignment systems. So when you deal with genomic data, you often have to do like either semi-global, global, or like partial local alignments of long sequences. Super common in omics. We will start with global. And as opposed to the common approach of computing the Wagner-Fish algorithm for the Levenstein distance or the Needleman-Wunsch for 2 strings, which have quadratic complexity, or the bandit algorithms that do kind of reduce the scope of computation by checking only specific radius of the Levenstein distance, we'll actually focus on newer algorithms that have not been implemented in modern systems, and hopefully will reduce the average case to linear time. On frequent cases, this will result in 2, maybe 3 orders of magnitude improvement in the speed of string alignment, and hopefully will buy humanity a few billion days. That's it. Come join me.
Thank you, Ash. Sean, you're up with converting bio research papers into code. Margarita, you're on deck.
Hey everyone, I'm Sean. I actually want to go the reverse direction of what Jeremy described. Go from papers to code. So Jeremy talks about turning projects into research papers, but I think there's actually a lot of value in archive preprints. Whenever I look at these, there's always these cool ideas, especially around the biosoftware realm. I'll take one example. I looked at the research agent paper, basically being able to intuit new problems and design experiments using agents. But it just has diagrams, it has a description of what it does, but it doesn't have the accompanying code. How can we convert research papers into code that's usable? I think this could be applicable to bioinformatics workflows. It could be applicable to things like the research agent, but it could also be applicable to a number of other protocols. So if you're excited about turning research papers into something that's usable, specifically archive preprints, come find me. That's it.
So Margarita is up. Stephen, you're on deck.
Hello everyone, my name is Margarita Gueleta. I work at the niche intersection of genetic genealogy and my academic research in population genetics and AI. And nevertheless, that population genetics and genealogy are hot topics in I'm sorry, let me show this. Currently, I'm working on a genetic kinship simulator, genetic genealogy is not a really known topic, but I feel that it is used in many cool cases solutions. I'm sorry. This is such a bad. Let me start over, okay? I was going to say. Have you ever heard of Autherm? Autherm is a startup that is using genetic genealogy for solving cold cases. And since 2018, many cold cases have been solved with combining genetic genealogy and population genetics. And I'm working on this simulator that uses founders. I'm sorry, I'm messed up today.
Stephen, do you want to talk about simplifying healthcare and bio document analysis? Stephen? Jim? Step up. I don't know if Stephen's around. So. Okay, Jim and Anil on creating a RAG-based LLM app to answer questions about the Cofounder approaches.
Okay, so I have good news and bad news and good news. The good news is that the best longevity drug in the world is called Klotho, K-L-O-T-H-O. It's named after the Greek goddess of fate. It grows new neurons in your brain, it protects the neurons you already have, it suppresses tumors, it also promotes autophagy, which is cleaning junk out of your cells. There's many other pro-youthful factors. It's a great drug for promoting and prolonging youthful vigor. The good news about it is your cells make it naturally, especially your neurons and your kidney cells. The bad news is that your cells make less of it as you age. The good news is there are people working on ways to teach your cells to make more of it as you age. My project for today is to use a RAG-based LLM to investigate some questions about Klotho. Klotho was discovered in 1997. There are around 4,000 published papers that include the word Klotho in the title. It would be great to use AI to dig into some of these papers and answer some questions. Maybe around like what is the typical Klotho serum level for someone with heart disease, a typical level for someone with kidney disease. There's also an interesting intersection between cancer patients developing acute kidney disease that is underexplored, that may have something to do with Klotho, so we may investigate something like that. Love to have AI people and bio people on the team.
Yes. Okay, Mickey, talk to us about Tinker Cat Life. Leah Houston.
Hey everybody. This is kind of fun. So I don't know if you can see these things. They're just little parts and pieces, and if I shake them a bunch, sometimes what happens, just by putting in what's called Brownian motion, they'll self-assemble, and that's kind of interesting. And the notion is, this is an experiment that was created by Skylar Tibbetts. Oh, there it goes. It just assembled itself into a thing. That's how life works. And all of us are made of stuff. And Skylar has been exploring this notion, and we used to fund him when I was at Autodesk, into self-assembling. So not computer-aided design and then manufacture separately, but actually designing the assembly code into the actual materials and into the products. So the products self-assemble. And so the question is, if life self-assembles, all of you were made through this crazy thing called Brownian motion and self-assembly and energy and stuff, how might we help the next generation think about this? How might we help move from forever plastics and forever chemicals, this idea that we could manufacture all of our things and all of our places with stuff that apparently can be infinitely thrown away, and never think about ever worrying about the sort of infinite trash heap that we call the ocean, or the infinite amount of stuff we make. How might we help the next generation from 10 years old up actually see wonder inside of structural proteins, actually see wonder inside of how the world works, and start designing it? So I used to work at a company called AutoCAD and Autodesk, where we had something called Tinkercad. And I don't know, has anyone used Tinkercad? Few people? So about 15 million kids use Tinkercad every month. And you can design circuit boards, and you can design generative structures, and you can 3D print things and stuff like that. So what I want to do today is build the first signal the first example of a plugin for Tinkercad that actually uses structural proteins like silk and chitosan and lignin and other things and sort of encodes self-assembly. It just added physics. Now I want to add Brownian motion in physics, and we want to explore what happens if we could actually help the next generation think about living materials and think about those transformations, so that we actually think about not only assembly and then throwing it away, but assembly, disassembly, and transformation. That's it.
Okay. Leah Houston is going to talk to us about credential and identity management. Vincent, you're on next.
So I'm Leah Houston. I am an emergency doctor. I have a background in molecular biology. And, how are we going to actually take the things that we're talking about today to market? How are we actually going to get it into the hands of doctors and patients? And how are we going to know which patients need what? And so I'm working on an identity management system for doctors to get workforce mobility solved, which is a really big friction-filled problem. But we have that's kind of like a Trojan horse for the future that we're trying to build, where we have targeted treatment for individual patients, precision medicine, the ability to find the doctors and allow them to understand what these tools are and how they can use them. Right now, that whole process is broken because physicians are kind of siloed in these large health systems and things like that. So we don't have to hack on my project, but I would love to work with people who are interested in either I like the data labeling idea of the research paper thing that Steve, I think, was supposed to do if he's back, or anything in proteomics or genomics. So if you guys are working on that and want me to be on the team, let me know.
Okay, Vincent Alessi is going to talk to us about multimodal protein embeddings. Gwen, you're on next.
Proteins are the coolest macromolecular compound in existence.
Yes.
Fighting words. They can basically be consumed for our nutrients, like our smoothies this morning. They can be used to treat circulating cancers that basically can't be targeted with chemotherapeutic agents. They can basically be used for structural applications, such as glues, inks, polymers. However, how to actually design with these materials and actually put them together in these diverse contexts is not easy. There's a lot of different dimensions to actually how you can guide and basically pick the proteins that you want to design and you want to basically use in your use case. So what I would like to consider doing, and maybe I'll join someone else's project that's cooler like Mickey's, but is essentially creating a library of nodes and edge representations where we're actually putting as an embedding layer in each a different type of actual data that would be relevant to their interactions. Sequence, Go ontologies, thermal properties, synthesis. And there's a lot of these things that may or may not be available within the context of today, but basically through creating an architecture where you could then actually access that through networks, Gephi, etc., you could create salinity and saliency of how you can actually build these things together. So if you're interested in nerding out on proteins and how to use them in cool places, maybe talk to me.
That's fantastic. Oh yeah, those are terrifying. Yeah,
Have you heard of deep origin? What? Deep origin?
So
Yeah.
They're doing what exactly I'm telling you.
Do I need to press something?
They say something.
no, you're good.
You know
It's
better
taking
than
a minute.
Okay, not fighting words, I do think proteins are great, but the precursor is RNA. So I am writing code that has already worked, so this may not qualify for the hackathon because we're supposed to start from scratch. We're not starting from scratch. But essentially, what's been happening in the last year is there's been codon optimization such that we've extended the half-life of Moderna's mRNA from 3.9 hours to 20 hours. So in the future, we might be able to get rid of the LNPs, which is the fat packaging for RNA. So today, I'd like to work on codon optimization, which usually takes 6 to 10 hours down to 10 minutes. And the attempt to do that is with linear parsing or cubic parsing. So if folks understand biophysics of RNA or have experience with linear parsing or cubic parsing, I'm wearing orange, so I'm easy to find.
Can I put your slide up?
Oh yeah. I had a slide. I had one slide. Anyway, so I'm showing our primary cell line results.
Are you predicting tertiary structure?
So that's already been done by another model of ours. So this is just one sliver of another model we're trying to build. But yes.
Cool.
Yeah.
Cool. Karen Sachs on DeepNet models of your biocircuitry.
Does my slide appear there?
So John goes through. We need access to your slides.
Oh, shit.
Do you want
Does somebody else want to go before me?
Sure. Yeah, Kadri, you're good.
Yeah. Hi, I'm Kadri. I went and put on yellow because someone right here before was wearing orange. Okay, so I'm a software engineer at Microsoft last, work in DevTools, trying to break into health. I'm founding in, I want to build is an AI coach that helps you manage your diabetes. So it'll take in all of your wearables, like your Oura Ring, your Apple Watch, your continuous glucose monitor readings, if you have them, and photos of your food, and help you like understand what you need to do day to day. And with the goal of getting to precision medicine on your app, you know, it should really be your like second brain to manage diabetes. We don't have to hack on that today, but if you're interested in this space, come and talk to me, and I'll find you if you're in a similar space. But yeah, that's kind of what I'm doing. Awesome. Yeah.
Thank you, Kadri. Karen, you're up.
Hi, I'm Karen. I'm a computational biologist, and my interest is I'm really in the middle. I'm a translator between the two fields, and my interest is in understanding the circuitry that runs your biology. So, you know, for those of you who are not that familiar with biology, inside each of your cells, there is a very, very, very complex circuitry, just like there is that flies the plane while your pilot is sleeping. It has a bunch of feedback loops and detectors, and it makes decisions constantly about what should happen in your cells. And when it makes the right decisions, you're happy and healthy, and when it makes the wrong decisions, you're screwed and you get a tumor and you die. And so we want to understand that circuitry, and we want to be able to simulate it so that if something is going wrong, we can predict which perturbation would fix it. In the context of cancer, we want to stop the proliferation. In the context of longevity, we want to look at the way your system is running and to predict, for example, what would reduce inflammation, for example. And that's going to vary in each individual person. So this paper that was published that has nothing to do with me, that I did not author, is a DeepNet model that takes kind of a generic large data set and predicts the effects of perturbations on the biological system, on the state of the biological system, the expression of every gene. And if we take molecular data from an individual person, which is very feasible and not that expensive, then we can say, what would happen in the context of this person, in the context of this person's circuitry? What happens if I make them eat more of this metabolite, be more protein heavy in your diet, versus make them take this particular drug or something like that? How does that influence the end effect of the cell states? So the idea would be to take this model and tweak it so that we can make personalized medicine.
Amazing, Karen. Amy Carl. Who you'd like to collab with in synthetic life. And Ainur is on deck with lab automation and robotics.
Hey.
Okay.
Hey, I'm Amy, and I just added my talk right now. So bear with me, because I'm just pulling up some stuff. I didn't realize that was an option, but I didn't want to miss the opportunity. I'm here today to offer what I am passionate about and look for other projects too that I can add value to. So I'm going to tell you a little bit about myself and the projects that I'm looking to work I am a biodesigner and bioartist. I've been working in AI and biology since like 2015. I was one of the early people to talk about that AI is not just about the technological, but about the biological as well. I'm a visual person, and the way that I do my research best is definitely very much scientific process, and very much I don't know the outcome of what I'm looking to learn. But I use the tools. I physically use the tools that I'm questioning, and I often use the body. I look at the implications of our emerging and exponential technologies on the body and on evolution. And while I'm working through that process, you just learn so much about what can happen. At that point, there's an opportunity. There's an opportunity to use our tools differently, redesign our tools in a different way for the preferable futures and the outcomes that we want. So that is the space that I work in. And really, this came out just earlier this week. My place that I feel really excited to work in is that kind of feeling when you have something on the tip of your tongue, you don't quite know the word. When you have something on the tip of your mind and you're like, I'm almost onto something. That is my sweet spot. That's where I love to explore ideas. And today, and what I'm really known for is projects like using generative design to make 3D printed scaffolds for stem cell culture into bone, redesigning vasculature for the heart through generative engineering and generative design, and then of course getting into bio. A lot of the local tech companies sponsor my work. This was work done. HP Labs has been sponsoring my work now for like 8 years, worked with Autodesk. I work with NVIDIA, a lot of big different companies. And today, some of the things I'm interested in working on, but I'm also open, is concepts in synthetic biology. In synthetic life. So how can we use our AI to help us identify and design synthetic life that then we can go test and build? But before we test and build it, also use AI to help us determine potential outcomes, both positive and negative, of the implications of building that. So not just building the synthetic life, but thinking through a larger life cycle of what could be the implications of that. I'm also interested in, I didn't have a picture for this. Yeah, I'm also interested in exploring like once in billion year events and how we can use AI to help us identify and determine what those could be in terms of fire. So thank you. So come find me if you want to work with me.
Amazing art, thank you. So Anne is going to talk to us about lab automation in robotics. Thomas, you want to?
I just added myself 1 minute ago. Hello everyone, my name is Ainur. You can call me Anne. I'm a lab automation engineer. Right now I started my own startup in lab automation. I did a little bit about myself. I did like bachelor's in mechanical engineering, robotics. I love robotics, all these tiny little projects. I worked at Genentech in Oregon as a in MSAT as a process engineer. I was working on this drug inspection semi-automation line. Then I worked at Garden House, biggest liquid biopsy company, with all these tiny little machines. I'm specialized in stars, Hamilton stars. Does anyone know what Hamilton star is? Okay, perfect for people. Basically, everyone's like COVID test, blood test, 23andMe, everything goes through stars. Basically, I know why they fail and everything. I fix them, I program them, pretty much everything. I was also a lab automation engineer at Personalis, worked with Agile and Bravo and other hires like Mantis. What I specialize in is method development. For example, just talking to scientists and programming robots and releasing and automating and scaling laboratories. I work on many different methods with scientists and I understand what scientists say. Right now, my goal is to find, I'm building a company, but also like I'm trying to do small projects today on like cell counting and colony counting. So I was wondering if I can meet more scientists, more machine learning engineers, learn more about synthetic biology, computational biology, and get probably try to publish a paper today. Thank you.
Very nice. Thomas Van, talk to us about speeding up clinical trials. Benjamin, you're on deck.
Hi everyone. My name is Thomas.
Not for you.
No. Sorry.
It's for the previous people.
Hi everyone. My name is Thomas. I work in the clinical trial space for more than a decade. And what I see to this space is very mundane and very manual labor entry. So it's actually very expensive. Drug from discovery to approval could cost around $2 to $4 billion, up to 10 years from that for the approval. So this is very heavily on our system, healthcare system. So one of the things that I really want to do is just how to speed up clinical trial using LLM and AI. One of the idea I have is, to optimize the clinical protocol. So every single clinical trial requires protocol to run a clinical trial. And a lot of time where is that, you don't get the right eligibility criteria, you don't get the right endpoint, you don't get the right assessment. So you have to redo the protocol multiple times. That's a lot of money to retrain the site, you have to redo the lab. So, right now we saw a couple of things that Omniscience was presenting. You can pull data from the science database. There's a lot of data out there from the FDA, from insurance. You can buy those data, and you can build better clinical trial protocol. And so that's something that we can utilize AI to kind of speed up.
Very nice. Benjamin, you're up on personalized longevity with deep models.
No.
I think I accidentally.
Mine's on.
Oh, okay. Sorry, I see. Let me open yours actually. Is yours on that subject?
No.
I'm switching.
But it's this one.
I see it. I'll just
Present mine.
Let's try. Okay, so millimers are short sequences of DNA that don't exist in the genome of a species. These are useful because they can be used to tag materials, so that that they've been tampered with in a lab, like from forensics. They're useful to potentially fight cancer. There's some promising cancer drugs coming out of these, and when looking into them, I found that there's really a lot of opportunity to optimize discovery of these millimers. So I'm not a bio person, but I want to collaborate with people who either to improve these discovery of these, or to apply the idea of millimers to someone else's project. I think someone was talking about identity management. That's something that could be useful for. So yeah, anyone who's interested in I guess algorithmic optimization, or anyone who knows, I'm not again at all a bio expert, so just being able to meet people who know more about that space is going to be very useful. So, yeah.
Allison, talk to us about citation malfeasance.
This just came to me.
And Jeremy and myself, you are on that.
Yes, okay, good for you. Hi, yeah, so just thinking a little bit more from the sort of research paper side, I love hearing about all these kind of tools that can help speed up the pace of science and get papers out there. I think something that raised for me is something in our current model that's already kind of breaking at scale is like peer review and actually having it be effective, especially when it requires like a level of detail that I think not all peer reviewers doing. Specifically, I'm thinking of citation malfeasance when someone cites a paper in support of a fact that they've put in their paper and it does not actually support that fact. We also see this in the media a lot as well. And you have a lot of people who they get autistically obsessed with something and they actually read the paper and all the citations and they're like, hey, you cited 3 papers in support of this statement and none of them actually did that. I think that's something that we could automate, that LLMs could be really good at with that kind of thing, and I could see a future vision where we bring in all sorts of sort of study, review, and criticism stuff, and we can also call out things like, sample sizes or p-hacking or whatever, or they tried to replicate this and it didn't, almost like community notes for papers could be cool. So that's sort of the thought. I'll be with Kajari in the yellow.
Very, very cool. So Jeremy Nixon is up with research agents. Jen Zhang, you are on deck.
All right.
You're on deck.
Oh, nice.
Okay.
Okay, sorry.
Okay. So at a previous hackathon, a friend of mine, Shira and I built what we called ResearchGen.ai. It's basically a site that lets you upload your research data. This process is super simple. It's just, you choose a CSV file to upload, and you give it your email address, and it emails you a generative agents process where it created experiments, it created evaluation metrics for those experiments, and then it creates visualizations and tables and writes the whole thing up in a research paper for you to read as a function of your data source. Now this is kind of crazy, but it's also super simple, and it doesn't give you control. So you can't currently say, make the research about X, Y, or Z, or do experiments that do X, Y, or Z. I have a variant that does this, but I guess the code that I really want to write today is some more like biocentric research agents, and really sort of flesh out how, given a specific data set and a specific bioresearch task, currently I'm planning on using the EEG data from a brain-computer interface data source, but I'm open if anyone else has a data set that they'd like to do this on, to try to get a clear research result out of this BCI data. So the research agent needs to come up with some clearly new knowledge that is correct and verified by this generative process. So if you would like to automatically sort of create new knowledge from data, let me know and we can code it up together.
Hey everyone, my name is Jan. I work on something very similar, and I'm a UX designer working on a clinical trial. And a lot of the data that we get from the lab is very poorly formatted. There's like comments everywhere in the spreadsheets and the CSVs. There's like machine-generated data, but also there's information on like what machine you use, just like things that are not the data get into spreadsheets all the time. Or your bioinformatician starts coloring cells, labeling things with footnotes in your spreadsheets, and that'll really mess up your data ingestion process. So what I'm working on now is using AI to try to clean your spreadsheets, right? Can you extract all the stuff about the data separately, and how can you identify on a spreadsheet? Sometimes you have a control group, sometimes you have other experiments all side by side, mix up together. How can we extract all that and label that properly, annotate it properly, and then send that to your database in a way that you can then feed to your other processes downstream. So if you're curious about that, join me.
Very cool. Okay, last two hacker lightning talks. Priyanshi talked to us about identifying gaps in scientific discovery. Nikhil, you're on deck.
Hi, I'm Priyanshi. I am the lead ML scientist at Gabby, and I build AI for breast cancer risk prevention and generally detection. And I don't have a PhD in clinical research, but a big problem when I started out was figuring out and formulating the research problem. And of course, it's not out in the open that these are the problems in this area. So my goal today is to build something that will help formulate a research problem in a specific area. And because I recently did this with breast cancer, that's the validation I'm going to use. So anyone interested in doing that with me, welcome. Talk to me.
Okay, Nikhil, talk about enhancing FDAI agent. And I guess last but not least, we'll have Ramesh on human clones.
Cool. Hey everyone, this is Nikhil. In the past, I used to work at Microsoft Research building ranking systems, but the last 2 to 3 years, I was majorly focused on using machine learning models for measuring aging, biological aging at full body and organ specific level. But in today's hackathon, what we wanted to do is we had open sourced a lot of agents for clinical trials and also genetics and drug age and animal aging, and all of these are RAG-based systems. These are all done in the previous hackathons. So in today's hackathon, we just want to combine all of them with a very clever router, and we want to have a voice modality to be able to talk to it And this is also going to be part of a larger open source project, which is called FDAI, which is a decentralized FDA agent that does automated research specifically focused on causal reasoning for clinical trials. So by combining all of these agents through a voice mode, we would be able to have better way of getting more data into the FDA agent, as well as people could get answers to any questions on genetics, clinical trials, and drug discovery to the extent the research literature supports. So it's going to be a very simple wrapper of router today.
Alright, hey everyone, I'm Ani or Anirudh. I'm from ETH Zurich actually. So I'm a grad CS student. I don't really have a background in bio, but one thing I was really inspired by was the Reid AI demo. Have anyone seen it? No? No. Reid Hoffman, he did like an AI clone of himself.
Yeah.
Yeah, and so I think it would be really cool. Like I'm in a phase where I want to figure out things and I have a lot of questions, and being able to just clone someone who's famous, who's got a lot of advice, like Andrew NG, and just call him up on WhatsApp and ask him questions. And the idea behind this would be sort of training it on all the video content, podcasts, books, anything that's written, and then putting this in like a RAG system or directly in the context, and then being able to have a voice chat with them on WhatsApp or something, training like an 11 Labs model. So yeah, looking for anyone to work with. Also another project. So
want to be interviewed by a digital version of myself? Oh, great question. Reid AI, introduce yourself to everyone. Hello everyone, I'm thrilled to be here today. I'm an AI-generated version of Reid Hoffman, his digital twin. My thinking and everything I am saying comes from a custom GPT that is based on 20 years of Reid talking and making content. So I've read all his books, watched his speeches, and listened to his podcasts. I think it gives me a pretty good sense of how you approach the world, Reid. So first, I'm going to challenge Reid AI with some questions to see what it can do and how good its content is in mirroring what I might say.
You did. Okay, so that concludes the lightning talks. Congrats to everyone who okay, so we are all, here it is, let's see, 1:20 p.m. Within 7 hours, the hope is that you have all created something incredible that you'll show off at the end of the day. So moments brimming with potential. Come up with your creative ideas, make friends with the people you just met, and we'll do something that we hope is incredible. Some context, so this is a 4-story mansion. We have 2 floors below this floor. They're all for you guys to hack on, so feel free to go down. Do not go upstairs. That's where our residents live. It's private, so please keep it to the lower floors. There's a bathroom here and on the bottom floor. I'll be pinging you for food, so when dinner shows up, I'll let you all know. If you're not in the Slack, get in the Slack. That's where we'll be sending out all of our messages, so come talk to me if for whatever reason you're not there. But basically, let's make this happen. So I guess personally, I have a strong sense that artificial intelligence is going to be able to accelerate bio research dramatically. And it's clear that there are so many dimensions on which that research frontier needs to accelerate if we want to solve essential problems. So in my mind, it is events like these in this direction that is really the highest hope for humanity when it comes to really transforming the way bio research is done, because by default, all of us are dead, and if we don't do something about it, it's going to be an unfortunate reality. I expect superintelligence will help, but it won't be trivial to use higher intelligence to make progress. And so this is one of the first sort of interstitial events, between AI progress with a bunch of AI hackers, AI PhDs, and bio PhDs. Everyone, heavy with youthful energy, coming up with new ideas and seeing what's possible. There have been like a bunch of launches over the last year of projects that are incredible. Even just this week, a team created a custom language model that was outputting variants of CRISPR. And they released this open CRISPR data set, and that gives people who are interested in genetic engineering a whole body of new proteins to play with. So there's incredible potential to come up with a project, launch that project, and potentially have a huge amount of impact on the future of bioresearch. Good luck. Let the hackathon commence.